overlapping the myosin, bringing the Z disc together and shortening the sarcomere, causing the whole entire muscle cell to shorten, putting tension on either bone at either end that the muscle is attached to. Right? Well, right now, that myosin is still attached to the actin. It's in this form like this. That's not enough to create much tension. As we were talking earlier, what actually happens is here's one myosin, here's another one, and they do this, let go, let go, let go. There's thousands of them. Remember that previous picture we showed?
What causes them to let go and reattach?
Such excellent questions you have.
Reading off this.
We practiced this before lecture. So, the myosin will not let go of the actin until the ADP is replaced by ATP. ATP can no longer, well actually take a picture of this because I want to erase and start all over. You guys will be able to see at the end. So if you want to take a picture, do it now. And then I'm going to continue starting at the top of the board. So remember, this was attached to the actin at this point. The muscle cell is not relaxing. The muscle cell is continuing to create tension. So just because the myosin head comes off doesn't mean that tension is removed because another myosin head is still attached to it. We're just kind of syncing that together. So this is where we started with our actin. Now we're here. I'm kind of redrawing this last diagram. So to get that to dissociate, we have to have a fresh ATP molecule. When we bring in a fresh ATP molecule, in this position, it's with the head like this, it's easier, the ADP comes off. But with an ATP on there, it no longer can fit on its binding site. So with ATP, it dissociates. So the myosin head released from actin when bound to ATP. But remember what that myosin head is? An ATPase. So it's going to break down the ATP and convert it back to ADP. And what does it happen with what does it do with the energy that's released? It goes back to its caught position. All right? So it's going to return to this position using the energy sorry, it should be ADP. From the ATP molecule. So that is what happens, back to your question, Catherine. That is what happens. So an actin binding site is available. This attaches to it, swings forward. ATP attaches, it lets go. ATP is converted to ADP. The energy recocks it. The calcium is still in the picture. A binding site will be available, and it does it again. So each myosin head keeps doing this and creating the tension.
Along the length of the muscle, does that happen
with every sarcomere?
All at the same
time.
It doesn't kind of just ripple down the cell.
we have, those transverse tubules are going in the entire length of the muscle cell, so it all happens at the same time.
Simultaneously along the whole.
All right, so notice that the myosin head will not come off of the actin. It will stay in this position until an ATP molecule comes along. No ATP? No, the muscle myosin head doesn't come off of the actin. This is what happens in rigor mortis, all right? Which means the stiffness of death. So depending on the body temperature, surrounding environmental temperature, anywhere from 5 to 12 hours after a person dies, calcium ions during that time are diffusing out of the sarcoplasmic reticulum. Why? Because they're in high concentration and it takes calcium pumps to keep them within the sarcoplasmic reticulum. All right, there's leak channels that are present there too. Without ATP, because the person's mitochondria no longer have oxygen to produce ATP, all right? So without ATP to man those calcium pumps in the sarcoplasmic reticulum, the calcium ions leak out. The cell doesn't know, I mean, a calcium ion's a calcium ion. Doesn't matter whether the cell's alive or dead, as you'll see tomorrow when we use the rabbit muscle. So the calcium binds to the troponin. The tropomyosin binds to its binding site. As long as there's ATP, it does this. But eventually, it's going to use up all the ATP in the muscle cell. Mitochondria aren't producing any more. And so when the last ATP molecule is used up, there's going to be a lot of muscle heads still attached, myosin heads still attached to their binding site on both flexors and extensor muscles. The joint becomes rigid because the muscles are in that contracted state. Not because a nerve triggered the sarcoplasmic reticulum to release calcium, but because they leaked out because the pumps weren't there to keep them in. And there are no new ATP molecules to allow this dissociation to occur. Okay, that has to be, ATP has to be present.
So what happens when you get cramp?
Too many calcium ions?
Huh?
Too many calcium ions or dehydration inside the muscle cell itself.
When they say they fall out of rigor.
Okay.
So that.
So we talked about this really, really early in the semester when we were talking about cell organelles. So if I'm not letting go of this, and I'm holding on to this tightly, and I'm pulling onto it, so then again, depending on temperature, if it starts at 5, then maybe 8 hours later. If it starts after 12, then maybe 20 hours after death, you'll start to see a disappearance. If you find somebody 3 days after they die, they're not going to be in rigor mortis. Okay? Does that mean that the myosin heads are no longer bound to actin? Proteases have leaked, exactly right. Proteases have leaked out of the lysosomes because they don't have sodium potassium pumps keeping the pH in there acidic. And as they leak out, we talked about this, somebody pulling on a rope. All right, so if you're pulling on the other end of this stick, and I can't let go and he can't let go, but someone comes along and breaks the stick, our tension has disappeared. We're still holding on to our little piece of broken stick, but there's no tension left. So it's the proteases breaking down the actin and myosin proteins that release that tension. So this is essentially the physiology of muscle contraction, all right? Now, as I indicated, to get the muscle to relax, we have to go in reverse. We have to get the calcium back into the sarcoplasmic reticulum. Well, that's not going to happen as long as the voltage-gated channels are open. And the voltage-gated channels aren't going to stay open as long as actin potentials are traveling down the transverse tubule. And the actin potentials are going to continue as long as acetylcholine is bound to the ligand-gated receptors and allowing sodium ions to depolarize the muscle cell. So the first thing we have to do is stop the lower motor neuron from releasing acetylcholine. And you can do that by consciously deciding you want to relax that muscle. Then we have to remove the acetylcholine that is there. And how do we do that? Nope, not with acetylcholine. Acetylcholine we do with acetylcholinesterase, which, like with the neuron, is found in the muscle cell. In our synapses, it was found in the postsynaptic neuron. So it's going to break down any of the free acetylcholine. So as this gets broken down and this dissociates from its receptor, there's fewer and fewer to bind to their ligand-gated channels, so they stay closed. that means our threshold potential drops, and then our action potential ceases. And once we get to the muscle cell, these voltage-gated channels will close. And now the calcium pumps in the sarcoplasmic reticulum will start pumping calcium back up their gradient to storage in the sarcoplasmic reticulum. So for relaxation, we have to remove that calcium. And so those are the steps of relaxation that you see on page 24. Notice it requires ATP.
So why has it been like, let's say you grab something and hold on to it, but if you relax, you're not like saying extend your arms, but it'll still like pull your arms out. Is it the same mechanism that's happening?
Well, we have joint muscles, I'm not quite sure what you're asking, but we have muscles on both sides. So I can relax this muscle, or I can contract the extensors to open it up. So for someone to pull on that, I have to relax both the flexor and, kind of, that's called passive range of motion, where you can't do it yourself, but someone comes along and moves it for you. So for passive range of motion, both muscle sets on either side of the joint have to be relaxed, or it becomes a stiff, jerky movement. Okay. Now, looking at all the events that we talked about for contraction and relaxation, and again, these are summarized on page 26, 27, 26. All right, so a summary of all of those events are on that page. That's a lot of activity going on in the cell. All right, from upper motor neuron to lower motor neuron to the actual muscle. So watch what I'm doing with my hand. As often as I do that, as quickly as each of these is happening in flexor muscles and extensor muscles, I have to relax my flexor muscles to allow my extensor muscles to do this, and then I have to relax my extensor muscles to do this. So as rapidly as I'm doing it, all of these events are occurring in those muscle cells. From thinking about doing it to sending the current down my upper and lower neurons to allowing the calcium out of the sarcoplasmic reticulum to pumping it back in again and pumping it back out and pumping it back in. As fast as that. Okay? I mean, it's even faster than we can think about it. All right? Now, a good way to practice your understanding of this after you've diagrammed it and you've gone through and you've seen what happens is to take the steps on page 26, photocopy that page or blow it up and cut it into strips, and then see if you can put them in sequence. Okay? What's the order of what has to happen when? Kind of mix them all up and go, okay, well, no, this has to happen before I can get the neurotransmitter out of the synaptic gluton. I have to have a synapse. I have to have an action potential there. So that's a good way to practice that. All right. This is a good place to stop. We'll pick, I promised, an hour and 15 minutes. So we will pick up the motor units. We'll pick up twitches and all the non-responses, TREP, tetanus, all of that tomorrow. And we'll talk about why when you eat your turkey it has light and dark meat. So you can espouse on this, expand on this at the Thanksgiving table.
I would totally do that too.
All right. So lab quiz on the muscle structure and the sarcomere components tomorrow. All right. If you need to continue working, or if you want to continue working, I'm sure most of you still need to, on the take-home exam, you may work on that. The PhysioX lab as well, assigned for Tuesday. We'll work on part of that muscle, not the PhysioX part, but we'll do the glycerinated muscle experiment tomorrow.
Is that the same thing that happens like when you get raw calamari or like soy sauce? Sometimes with
Yeah, soy sauce has all that salt in it.
yeah,
It's polarizing.
like freak people out of doing that.
I've never seen it, but yeah. Tuesday?
Check them out. Look, you ever see black hair like those
Yes.
tell me. Sometimes I can't see them. I want to have like blonde ones that come out
Uh-huh.
here, and they like these seems like it's about like this long, and I'm like,
No one's gonna, that's what I was gonna tell you.
There's, you have to look at both nerve and muscle. Sometimes you get a muscle twitch. Your eyelids get something. That's nerve fatigue, where it's, allow it, spontaneously releasing neurotransmitters. You can have spasms with too much calcium inside.